Welcome to an overview of drug-receptor interactions, where we'll explore the mechanisms, kinetics, and therapeutic implications that guide modern drug design. In this presentation, we'll link basic binding concepts to practical decisions like dose selection, antagonist strategy, and personalized dosing, providing a roadmap from molecular recognition to clinical impact. This slide contrasts reversible, non-covalent forces, like hydrogen bonds, ionic bridges, hydrophobic pockets, and van der Waals contacts with irreversible covalent bonds, and explains how those interactions set affinity, reversibility, and therapeutic behavior. From the diagram, you can see non-covalent binding is dynamic and tunable, while covalent links produce long-lasting inhibition with distinct liabilities and optimization challenges. Here we examine drug-receptor equilibrium and the key metric Kd, which quantifies affinity at steady state. A lower Kd means tighter binding and stronger interaction, while a higher Kd implies weaker binding. The figure links these values to fractional occupancy and shows how Kd helps compare ligands quantitatively and informs dose and occupancy decisions during development. This page explains how receptor occupancy relates to pharmacologic effect via a hyperbolic curve. Response rises with more occupied receptors until saturation limits further effect. The slide also highlights receptor reserve, where full agonists can reach maximal response without full occupancy, whereas partial agonists produce sub-maximal responses even at saturation, shaping therapeutic expectations and dosing strategies. We categorize agonists by mechanism and outcome. Full agonists elicit maximal system responses. Partial agonists give sub-maximal responses even when receptors are saturated. Inverse agonists reduce constitutive activity, and biased agonists preferentially activate selected signaling pathways. These distinctions matter because they influence efficacy, safety, and the opportunity to fine-tune drugs for fewer adverse effects. This chart contrasts competitive antagonists, which shift dose-response curves rightward but preserve maximal efficacy, with non-competitive or irreversible antagonists that lower the maximum response and flatten the curve. Practically, higher agonist doses can overcome competitive blockade, while irreversible antagonists require alternative therapies because dose escalation cannot fully restore efficacy. Receptor activity reflects a dynamic conformational equilibrium between inactive and active states, and efficacy arises from how drugs shift that balance. Agonists stabilize the active state. Neutral antagonists bind both states without preference, and inverse agonists favor the inactive conformation, reducing baseline activity. Understanding this helps predict drug effects in systems with constitutive receptor signaling. Allosteric modulators bind distinct sites to change receptor conformation and modulate orthosteric ligand activity, and the slide lays out positive, negative, and neutral modulation. Allosteric approaches offer subtype selectivity and novel therapeutic mechanisms by enhancing or reducing orthosteric responses, expanding drug discovery opportunities where traditional orthosteric selectivity is limited. This slide summarizes essential in vitro assays for measuring binding parameters. Saturation binding yields Kd and Bmax to define affinity and receptor number. Competition assays determine key and relative potency of unlabeled compounds. And kinetic binding measures association and dissociation rates, KO and COF, to understand residence time, all critical inputs for SAR and target validation. Quantifying potency and efficacy uses metrics from concentration response curves. EC50 defines potency as the concentration for half maximal effect. Emax indicates maximal achievable response and clinical benefit ceiling. And PA2 estimates antagonist strength from the shift in dose response. These parameters inform therapeutic dose ranges and feed into cross-system modeling for first-in-human predictions. Drug receptor selectivity is key to minimizing off-target effects and widening the therapeutic index, and the slide outlines the challenge of nonspecific binding. The solution involves guided iterations, using structural biology, binding kinetics, and medicinal chemistry to design highly specific drugs, ultimately reducing adverse effects and expanding the safe dose range for patients. Pharmacogenomics integrates genetic information to optimize drug receptor interactions by revealing polymorphisms, splice variants, and expression changes that alter receptor density and coupling. By combining patient genotyping with quantitative interaction models, we can predict dose-response variability, maximize efficacy, minimize toxicity, and move toward individualized dosing regimens tailored to diverse patient populations.